Hey, Xiling, thank you for coming in today. You have just a really outstanding, profound career, definitely impacting the world in many, many dimensions. Uh, I can see you really making so many continuing contributions. So thank you for coming in to share some of your insights with our audience today.
Stephen, thank you very much for the invitation. I'm very delighted to join this interview with you.
Xiling, one of the things that my audience is always curious about, and my audience consists of scientists and investors and CEOs, it's pretty broad. It actually tends towards people who are more in the executive suite. But, we're always curious. You have this outstanding career to this point, and it's going to continue. What were maybe one, two, or three inflection points that made this sort of wonderful, marvelous person that you are today?
Great, thanks. Kind of think back, right? I have a kind of a non-conventional trajectory, had a kind of many several pivots that I think really made a kind of difference. And really till now, I connecting the dots and seeing how kind of important kind of they played in terms of where I am now. So if I think about the inflection points, I think there are several. Number one is at that time I grew up in Shanghai, and I did my high school in Shanghai, and back then I actually met with an admission officer who came to Shanghai from Grinnell College, and he said, "Have you thought about applying to U.S. colleges?" Right, and then he said, "Oh, you need to take SAT." I said, "What is SAT?" and kind of these kind of serendipitous, but again, kind of he described to me the how different the U.S. college system is and the freedom you have versus the Chinese system, which is very rigid, and you pick your major when you apply to college, and that's it, right? That's going to be your career. So long story short is I kind of end up doing a 3-day walk-on and took the SAT, probably the very first Chinese student in Shanghai took the SAT, and end up getting to Stanford and started my undergraduate career. So I think that was the first inflection point that really kind of plugged me into Bay Area. And from undergrad years, where I was a Mayfield fellow and really had a first exposure to entrepreneurship and kind of learning how translate academic research into entrepreneurship and startup companies. But my second inflection point is when I was at Cornell University. I started as an electrical engineering professor. I actually did a circuit design for my undergrad and a PhD. And at that time, I saw a paper came out from Dr. Hans Clevers in Nature in 2009, right after I started, in describing this new technology called organoids, where for the first time, you can grow tissue or tumor in 3D kind of gel 3D culture system that can mimic how they grow in the body. And at that time, I just felt this gonna change the world because for the very first time, we can really studying them outside a person's body. And also joining my background in electrical engineering, I saw the analogy between this technology and the Moore's law, right, where we shrink the device from vacuum tubes to transistors and really enable from the 1960s the big radios to this day, right, cell phones and computers. So I think and the same thing happened with sequencing. So I really had a pivoting moment, like, look, I can apply my knowledge, but really tackle a problem that biologists have not realized, which is how to scale this technology and to really make a platform like semiconductor, like next generation sequencing. So I kind of pivoted my lab and kind of got into biology, and that was my second inflection point. The last inflection point was when I kind of moved to Duke and directed the position center. I had my first inaugural center symposium where Dr. Hans Clevers was giving a keynote speech. And during the keynote, he was describing the technology, get everyone excited, but he said, look, right, this technology has several major challenges. It's too slow. It's, the reproducibility, and the ability to capture not just the tumor cells, but the whole environment, especially the immune, right? So these are these are key challenges that to allow this technology to be used in the clinic. And I have a VC happened to sit next to me, and she turned around, said, "Look, I just heard you're working on this over last dinner, and you guys should come to the Silicon Valley." And that was kind of we went to the Silicon Valley and got our first term sheet. So that's a third inflection point. So if you look at through those three, I think that my key takeaway is that first is serendipity does happen, right? And this usually happen in a big way that really changes your career. But second is you also had to be ready for it, right? I think at each moment, if I thought about it, I've been thinking about it, I was kind of ready, and I kind of jump on opportunity. So I think it's very important to kind of be ready, but also ready to switch direction and jump into an area that really gonna make maximum impact.
That's a really interesting journey, where you're in Shanghai and you get this opportunity to think about U.S. colleges, and then you go through this sort of process, and you you get into Stanford from high school, and you didn't have to write your national exams. Like in China, you have these national exams and things like that. So you're able to bypass all of that. But like you said, you were ready for that moment, and that's where you got your bachelor's of science, you got your master's of science, and you got your PhD degrees from Stanford University. And Stanford is ranked #1 in many areas of the world, right? So many areas of domains of science and technology. So that's a very, very fortunate. And then you mentioned, seeing this speech by Hans Cleaver, and you talk about this idea of organoids, and in fact, based on the earlier work, I guess, of Yamanaka, and this idea, and then being able to sort of coax stem cells, pluripotent stem cells, to become something, right? And just for the benefit of the audience, an organoid is like where you get sort of like a mini brain, or a mini a liver, or you get a mini heart, or so on. You can grow these cells so you can coax them to become, sort of replicate at least early stages of different parts of the body. In fact, they're getting more and more mature of how you can create these organoids. So, and you you're in the company of this gentleman that actually invents this new way of doing things, right? We can replicate almost like little mini me's and then you attend this lecture where he says, hey, there's all sorts of challenges though, and you're applying your system approach, your system knowledge, your electrical engineering background, and you're thinking, I can solve that. and then you just happen to be beside a venture capitalist, and that venture capitalist saying, oh my God, you solve that, you can make profound changes to the world. You can really accelerate the ability to do drug discovery. You can accelerate the personalization and the precision of cancer detection and how to manage that, how to target, and really condense the time required to do all of this, right? And by many factors. So not just a little bit, but by considerable. And you can scale it, maybe 1,000 times or whatever. I mean, I guess there's no limit really to scaling it. So, I mean, it just kind of incredible, this journey. Did I get anything wrong in my sort of reflecting on your journey?
No, yeah, no, absolutely. No, you got absolutely correct. I think, that's kind of how we, get into a new field and thinking about using the past knowledge, but you're bringing expertise from a different domain, right? A different way of seeing things, and coming in and kind of really, addressing a critical math need, right? In life science, people don't think in terms of scalability that as engineers we think about all the time, and platform building. But also, just for the audience, right? The way we scale this is by growing from a tiny patient biopsy and growing, tens of thousands of little mini me, as Stephen said it, or basically kind of can also imagine a little patient avatar, and in tens of thousands of little droplets right? And so now you may basically make so many copies, and then each one becoming a little avatar that you can do many different drug testing, but also in a record speed. We can do in the clinic within 10 days, right? The conventional organoid technology take 2 to 3 months to grow enough to do these different testing. So this really having a significant impact in the clinic, because no patients want to wait 2 months, right, to be put on a treatment. And now we can allow them to be on treatment in 14 days, the regular clinical flow.
Yeah, I mean, let's explore that. There's so much to unpack here, right? Unpack meaning, break this sort of really interesting ideas into its subcomponents so that we can understand the implications of that. So, for many in the audience, they probably aren't familiar with organoids and the fact that that was a major discovery and a major clinical tool and then you find, but before that, I think most people are familiar with doing tumor biopsies and then trying to figure out from those tumor biopsies a sort of a regimen of treatment. So what would be the time factor before organoids if you had some kind of cancer and then you do a biopsy, and then from that biopsy you then determine can we do something that's a little bit more precise and personalized and so on, and we call that precision and personalized medicine, right? So what was the time factor there? And then we see sort of this acceleration occurring, or at least a really more targeted approach with organoids. And then we have this more condensed sort of unlimited scaling capability with your droplets that you talked about, your little mini-me's, your avatars, or your what you call your MOS, right? Your micro-organospheres. So first of all, what was the time factor? And what a lot of people think is cancer, you do tumor biopsies and you try to figure out what's the time scale there.
Yeah, great, Stephen. Yeah. So absolutely, kind of for the audience, right? Kind of giving a little overview of, kind of why this technology matters. So, first, what's the problem, right? Right now, as many of you know, in the cancer field, when a cancer patient have a chemotherapy or IO therapy or targeted therapy, what happens is still the majority of the patients are taking a drug that doesn't work on the tumor, right? And anytime. And while the treatment progress, right, when you had the first line, second line, third line, the chance of the tumor respond to the treatment is less and less, right? The first line, maybe you look at about 30%. Second line is maybe 10 to 15%, and the third line is under 10%. So that means 90% of the patient on the third line is taking a drug that has a lot of toxicity, right, and doesn't really do much to the tumor. So the ideas of precision medicine is, can we be more precise in terms of finding the right treatment for the patient so that kind of more patient benefit from the treatment. The challenge is that, right, so that field has been driven by sequencing. And again, I directed the Duke Precision Medicine Center. So I was part of, right, also that camp, which is, look, if you can sequence the tumor and know the mutations, then you can find the right treatment, right? Problem solved. Okay, the biggest challenge is that even till this day, only about 10% of the patient actually benefit from the sequencing. The reason is the majority of patient, when they sequence the tumor, it does not happen to have a mutation that we have an existing drug to target that mutation, right? And that actually happens in a minority of the patients, about 10%. So the majority of patients, 90% patients, are still go on to have the standard regimen, kind of in a sense of shooting dots in the dark, right? Hope to hit a target. So so right now the flow is, right, you take a patient biopsy, or you kind of try to get from blood, you try to sequence their tumor, and if the patients happen to have a mutation that you can target, right, within that 10% lucky group, fantastic, you give that patient a drug. Otherwise, the patient go on to have, it's called a standard of care. Now, what this technology now allow, and the traditional what people try to do, but if you look at the conventional, right, people try to, it is a straightforward idea. Say, look, can I test the drug on the patient tumor, right, before I put it into the patient, right? That's, if you think about it, there's nothing, better than actually being able to do that. And the problem is the current technology, people have been doing this kind of patient-derived xenograft, which is try to take a little patient biopsy and try to grow in the mouse, right? These are immune deficient mouse, means the mouse don't have an immune system. So you're not going to reject the patient tumors and try to grow that tumor in the mouse, then doing drug testing. So the problem of that is, number one, oftentimes for many cancer type, the success rate is low, right? So you're going to get, maybe 30% of times you can grow a patient tumor in the mouse. 70% time you're going to fail because you start with so little materials, right, to try to grow in the mouse. But second, also the biggest problem is take about 6 months, right? You're going to grow the tumor in the mouse, and you put it into a second mouse, and you grow it again, and that's kind of ready. So now you're thinking about asking a patient to wait for half a year, and 70% of the time, it's actually end up not growing, right? So you can see this is not feasible for the clinic, and although it's great for research. A second approach is, the conventional organoids that my co-founder Hans Clevers and I co-founded the company Xiling, right? But before that, when he talked about a challenge, the thing is, if you try to grow from a little biopsy, grow into an organoid, growing these 3D structures and try to do drug testing, that still take about 2 months, right? To have enough cells to do even just a handful of tests you want. So these are the kind of clinical. So again, right, the patient cannot wait for 2 months. So you're looking at potentially a second line treatment, right, after the patient had a treatment. But at that time, when the patient received the treatment, the tumor also might change, right? So if you did it from 2 months ago, it might not represent what that patient's tumor at the time of the second line treatment. So these are very fundamental challenges, and also these processes are very hard to automate, right? Because these are all done by hand. It's very extreme. Oftentimes we talk about in the lab, right? You have a person who have a good hands, and they get results that are very different from someone, who have a different skills. So what Xiling does now is completely flipped that, right, constraint. So now from a patient taking an 18 gauge biopsy, which is a very small, little tiny needle biopsy that has no harm to the patient, and then which is standard anyway, but the patients take the biopsy for sequencing anyway. But then what we do is within 10 days, within 3 days, all these little patient avatars, tens of thousands of them are established from the biopsy, ready for drug testing, and also still with the entire, right, environment. So not just the tumor cells, but the stromal cells and immune cells, right? Recapitulate the whole ecosystem going on in the tumor. And then, usually by day 8, by day 8, we can finish the drug testing, so how the patient tumor respond to all the possible drugs, and even experimental drugs, right? Drugs that's in clinical trial. Because if the patient does not respond to the standard of care, actually they might benefit by, go to the clinical trial that, right, testing on a new drug that might work on them. So, then by day 10, the clinician is going to get a report of our results showing, right, what drug might work on the patient.
Right. So, that is, very fascinating in this idea that, from a 6 month and then, in the older sort of technique, which is still used in the world, and then you use these organoids and you can get it down to maybe 2 months, but you can't replicate the environment that tumor and cells existed. So it's not really a realistic sort of picture of what's happening in the body itself. And then, so from 6 months to 2 months, and yet there's all of these challenges, right? Because it's not realistic, picture of what's happening within the body. And then you've been able to encapsulate within these droplets, and you call them moss or microorganispheres, really the tumor, but also the surrounding sort of environment that tumor cells exist. And therefore, you get a very accurate picture, and that's why you call it an avatar, because it's really a representation of that person, and pretty precise representation of that person. And because it's microscopic, you can scale it. That means you can, create 2,000, 10,000 versions of it simultaneously. And you can apply all of these different sort of therapeutic regimens against it to see what works and what doesn't. And you can even test maybe combining different regimens together in a compressed time frame, as you mentioned, 3 days, 10 days, and so on. We're really within 10 hours where you start getting some idea what's happening. And by 10 days, you got a really good idea what's happening. You get a very good idea what's going to work and what's not. And if you stage a number of regimens together, you could maybe even have a better outcome. But you can do this in this environment where you do thousands of these things at the same time. That's the magic of your of what you've done. And you indicated that there's some kind of standardization, because you're electrical engineer. You're always looking at a system approach. Can we standardize this? Can we automate this in a way? So can you talk about that standardization, the automation aspect, and how do you achieve that, or how are you achieving that automation versus a manual process, one at a time, waiting months, and you're not replicating the environment? That's the person itself where you're able to do that. So how is that going to accelerate it, make things so much better?
Yeah, no, thanks, Stephen. You really captured the essence, right, of this technology. And also to your point, right, some audience might have the question, say, oh, why mouse can, right, be the first technology that really recapitulate the entire environment, right, including all the immune response, while the previous technology can't, right? Just kind of from a very quick point of view, if you think about, if you try to passage or grow these cells for 2 months, what happens is first, right, all the non-tumor cells die because they are not tumor cells, right? They cannot grow in the plastic. And the second is, you are really selecting a special clone that's the fastest growing in the plastic, right? So you are kind of you're putting artificial selection pressure to direct the evolution and picking out a specific clones that going to overtake the population, and oftentimes you not represent the original patient tumor, which is heterogeneous, right? You have many different kind of clones that actually grow in that environment. So to your point, right? So that's why this technology are really able to capture that environment, which is very important to predict whether a patient tumor are going to respond to the drug. But also second is what the epiphany moment we achieved. Again, that was also a serendipitous finding, right? When we first generated the technology, we're thinking about scalability, but we did not expect that the environment can be kept too well. But that was end up a really kind of, so that came out of a conversation I had with Dr. Vibra Gav, who was leading the Broad Institute, right, the Genomic Institute at MIT, but now she's leading Genentech. And she suggests us to do single cell sequencing, basically looking at what are all the cells in this mouse. And the result came out, right, just kind of shocked everyone, which is the entire environment still very well kept for a very extended period of time. And that came to the whole field as a kind of really big surprise, right? No one would have expected that. And that's why we want to cover a story of the Cell Stem Cell Journal, which is the top journal, right, about, or tissues and organs. And also National Cancer Institute basically send out our technology, the blog, to all its subscribers. But then coming back, and it's also, right, interesting. Now we think about it, is the previously, as engineers, and myself, bioengineers, we kind of went about kind of the wrong way, right? We try to recapitulate the complexity of the tissue by artificially making things very complex, right? We're going to have all these different cells. We're going to use bioprinting. We're going to put everything exactly where it is, right? Try to mimic, and then the cell die, right? You somehow just can't do it. But what we realized, but if you think about in the human's body, when a tumor is growing, it has all that, and all it takes is really just nutrients from the plasma, from the blood, right? I mean, you don't need to inject artificial cocktail into a patient's body to sustain all that environment. So what's happening here is that, again, coming back to the idea of the ecosystem, right, or the niche. Basically, in the body, if the tumor cells talking to the stromal cells, changing the immune cells, they create a ecosystem that self-sustaining. So what we end up doing a very simple concept that by putting them right into a little, the microorganospheres and allow them to very quickly talking to each other again and reestablish that niche, it actually becoming a self-sustaining little ecosystem, right, in the patient avatar, Stephen, as you pointed out, that you can do all these drug testing. So again, that's kind of the idea about, allow those, the environment to do its thing. And the environment actually can it kind of is an equilibrium that can, self-propagating. Now, to your point, Stephen, but then also making automation also very critical, right? Because to do high throughput, you obviously need automation, and that's very important because I think about where cancer treatment is going. Right now, now one drug is not going to do it all, right? We have to look at the combinations, and that's what every farmers and every clinical study is looking at. But the challenge is that, especially for drug-resistant cancers, that's what you want to cure, right, or at least control. But the challenge is, you can imagine that dimension or that space, exploratory space, become exponentially large, right? Because if you have 3 drugs, you want to test them, and that's already making a clinical trial impossible, right? If you want to try all combination of just 3 drugs. So and we don't have that many patients and to do this kind of trial. So a way using our mouse to test those is address a fundamental limit of how we can find the treatment for the patients. But to your point, now also making automation even kind of more critical, because you are looking tumor cells in its environment, right? It's not just tumor cells growing; you have all the other cell types. So what we allow us to do is really a very robust but also sophisticated system that combining both hardware and software. So what the hardware allows us to do is the fundamental property of mouse actually enable these high throughput automated system that conventional organoids or mouse is impossible to do. So by being able to capture these tumors in the avatars, and now when you're putting them in a plate, what happens is these patient avatars, because they are 250 microns in diameter, making imaging very easy to capture every single avatar, right, at one snapshot at the same focal plane. If you can imagine, right, this cannot be achieved, certainly not for mouse, but even mouse animals, but even for conventional 3D organoids, it's a 3D structure that, right, you have to be able to capture every single plane. But in our case, we make this automation very simple. And also these mouse are easily dispensable, right? It's very accurate dispensing. Basically means, you can use acoustic dispensing that putting exactly the number of, cells per mouse and MOS, and then, exact number of MOS per well, right? So this allows and putting to very high density well. So the hardware allows us to do this kind of high throughput manipulation by robotics that the conventional technology can't. But then there's a second piece is the software, right? Because when you scale up the numbers, obviously these cannot be done by hand anymore, right? It's going to take people's months to quantify every single patient avatar by hand. So the software does is using artificial intelligence, right? We are having the algorithm that go into every single mouse, pick out the tumors, right, that forming the tumor spheres, from the environment, right? Be very precise on measuring the tumor cells response, and also for the immune therapies, what immune cells are doing, right, to these tumor cells. So we have that kind of resolution to really be able to tease apart, right, within the tumor, when you're adding a treatment, what's happening to the tumor cells, so what's happening to the environmental cells, and having this and completely unbiased, objective, right, readout from the automated AI algorithm that kind of providing that quantification that's highly reproducible, right? Like you go to every single hospital partner we collaborate with, you're going to get the same readout, right? Rather than, hey, if you have a technician in this hospital versus that hospital, you're going to get very different readouts. So we standardize it, which is essential for FDA approval and also for basically payer reimbursement on that front, but also for drug development, because now you can imagine we have the very first system that recapitulates the entire patient, right, the tumor environment in these patient avatars. And another challenge for pharmaceutical companies is that, right, everyone complaining the drug is too expensive and take too long, right? What people don't realize is more than 90% of the oncology drug fail in clinical trials, right? And on average takes 6 years and $400 million. So you can imagine that using this technology, and now it's also very reproducible. This is also allow farmers to now take advantage of this technology to really pushing the next generation therapies and de-risk their clinical trials. And also the most important thing is finding the right patient population, right, that would respond to the treatment. So I think, if you even talk to farmers, right, people say, look, we know the drugs is expensive, but take a lot of R&D. It is okay if that works on the patient, right? But the biggest concern is nowadays we are developing more and more expensive drugs that might not work on the majority of the patients, right? And these are, we need a fundamental technology kind of solution to address this concern.
Yeah, I mean, you've used different terms like clones and things like that, but really what you're saying, and I'm just sort of translating for my audience, you're really creating, and you use this term recapitulation as well. So really what you're doing is you're constructing, and you can do this really, really quickly across tens of thousands. In fact, there is no limit. You could do millions if you had a large enough area, right? And these wells that you put these droplets in are tiny, so that's why you can basically represent the human being, a person within a droplet, and all of the constituent cells that surround that tumor that actually interact with, including the immune cells, and you can do this instantly across many tens of thousands of times, thousands of times. It's easy. And then you can actually watch the interaction of drugs and drug combinations because you can almost do it on a 2D, even though it's 3D, it's not an organosphere 3D where you got so many different slices. You can really do a 2D sort of analysis of this. And because you're using artificial intelligence, machine learning, it can do something humans can't do. It can say, this is what's happening with the tumor cells. This is what's happening with the surrounding cells that tumor exists in. And you can see the interaction that's occurring. And because it's so small, you have more surface area, basically, to allow more of this infusion of these different drug regimens. So again, much so efficient. And that's where you're saying with drug companies, when they typically do a clinical trial with people, that takes time, and you have to gather these people, and then there's the risks associated with it. How do you mitigate that? You could create these artificial versions, really clones, replicas, and they're so tiny that's why you can mass produce these replicas, these avatars. The avatars having clones of the different cells. And you got them encased in this little droplet, which you call a microorganosphere. So, and then you can do these mass drug combination testing as well, because at different concentrations, even 3 different drugs at different concentrations. What an impact! You can do this on scale and massively. Not just in one location. Can you imagine if every lab in the world did this, every hospital in the world did this, and you combined all of this knowledge, then you would get an exponential, you're already getting an exponential increase in the capabilities of therapeutics and diagnosis and so on. So, and then on the discovery side as well, right? With pharmaceuticals. And you're just at the beginning. It just blows my mind. I mean, I'm just amazed at what you've been able to do by combining this sort of system approach of electrical engineering with this biomedical side and the medical side, and this whole magic of transdisciplinary, right? Or interdisciplinary, where you're really having different aspects come together on some kind of a problem, and then you can solve it in really novel ways and produce amazing results. Okay, so in my little discussion, did I get anything wrong?
No, Stephen, no, you capture it very well. And also, also thank you for pointing out, right? Certain terms, jargons I use, like a clones and recapitulation, right? And but also kind of to elaborate on that point a little bit more, another kind of fundamental capability of our mouse that kind of the previous technology don't have is another, right? Another critical matter need in the clinic, which is a patient receive a treatment, right? The tumor responded and it looks great. After 3 months, they grew back, right? And become resistant to the treatment you were giving. So the reason is because not all, even within the same patient tumor, not all the tumor cells are the same, right? It's not completely homogeneous. It's almost thinking about, as a human society, we sometimes talk about why it's so challenging to cure cancer is because you are fighting evolution, right? When you put it on, the cancer cells are evolving, right? Over decades when they develop, and then when you give it drug treatment. What the another mouse technology what offers is, right, you can imagine now we have these little patient tumor avatars, and they also represent the different population from the patient tumor, right? So often what we can see is you add a treatment, let's say 90% of avatar gets killed, but 10% of avatar are actually able to kind of survive that treatment, right? And now oh, right, like what we can directly translate to the clinic, we say, look, when you're giving the drug, we know the patient is going to have a 90% response, right? It means the tumor is going to shrink to 90%. But you also know that 10% of the remaining tumor is going to grow back and they are becoming resistant. But what this technology can allow you to do from the very beginning, right? Either explore combination to say, look, can you adding a drug that also targeting those 10%, right? Rather than waiting for them to come back. Or another thing, say, look, okay, we have a treatment, we know going to work on 10%. If the patient comes back, you have a second line treatment ready. So I think this is also important, right? In the conventional technology, as I mentioned, you are asking them to grow for to a month and making the whole population right, kind of selecting certain population that don't necessarily represent this heterogeneity or diverse of the patient tumor, that mouse actually are able to address this very fundamental, right, limit of our current cancer treatment and hopefully bring much more benefit to the patient. And then also second to your question, right, is that as you mentioned, right, we still right now the FDA approval process, all that heavy rely on animals, right? But nowadays with the newer treatment like the targeted or the immune therapy, right? These newer cancer treatment, there's not good correlation with animals, right? So again, we are actually killing a lot of animals that sometimes also prevented a drug that truly work go to the clinic because there's no resemblance from the animal model and actually the human disease. So, and again, some of you know, right? There's a legislative effort now, actually, both, I think already passed House and Senate, right, on rethinking about this and changing these kind of, blanket animal requirement for any drug development. But now, so all the drug companies are, kind of gearing for that. But the question is, what is the alternative technology, right, that can help us really doing that much more efficiently and really kind of picking the right drug for the patient, right? Now, in a sense, there's almost a folly that if a drug doesn't cure the mouse, the drug will never get to the people, right? But the mouse is still very different from humans. And also many drugs actually work on mouse, and that's also right. Always the joke in the field is it's easy to cure a mouse, but the drug doesn't work on people, right? Both ways. So we are filtering a lot of the drug that might work, because they don't work on the mouse, but also letting a lot of drug pass through the mouse that end up not working the clinical and harming patients while wasting a lot of time and the resources. So again, for this technology, potentially these avatars and can really replace in a way a lot of animal studies that, really helping patient in a meaningful way?
Yeah, I mean, again, you went back to this idea that, a tumor is just not homogeneous. It's not just one kind of tumor type when it's in the body. It has many different manifestations of itself, some of which can be drug resistant, some of which can be, responsive to some kind of therapy. And that presents a problem. Whereas with these little avatars, which are really a duplicate, a micro duplicate of what's in a human, including all of the different kinds of tumor manifestations and the surrounding environment, then you can see what's happening, and you can say, well, why don't we do some kind of combination so we can knock it out totally, right? Mm-hmm. And or if you can't knock it out, then at least what are secondary lines of defense we can put into the patient. And because it's such because it is an exact replica of the patient, but at a microscopic level, then you can do it very quickly and safely and so on. And you can replicate all these different combinations. That's the scaling aspect. So that's the magic of what you can do. And then, as you talked about on the animal study side, animals are not a good representation of a human, and something that works in an animal won't necessarily work in a human, or something that works in doesn't work in an animal may work in a human, and it's so inefficient. Whereas if you do it this way, you get really a precise match, and again, that's the efficiency of your system. And because a lot of my audience are computing scientists, I just want to make sure that they realize that behind all of this, you've got computing science, especially on the AI side, the machine learning and so on. You've got computing science in the background, and then you also have the robotics side, where you can automatically, spin up all of these, microganospheres and have all of these drop these micro wells, and then the imaging systems, which are all automated and so on, right? So adding,
so Steve actually have a great point. Also, you're talking about the software side, right? So I think there's another aspect probably the audience, probably not aware of, which is, first, if you think about a company, actually, right, almost very significant resources on the software side, because all the software interface, all these new devices, but then also the data analytical side, right? Providing AI. And also you're talking about patients, right? So these data have to correlate with clinical patient outcome, right? Which is, kind of, I gloss over some of the complications, but as I mentioned, right, the patients are not binary, right? The patient, when you give a drug, it's not like, oh, I'm completely cured, or the drug completely doesn't work, right? So there's many shades of gray in terms of response, and how, so the patient avatars kind of really recapitulate that to give clinicians more information, right? Thinking about a more holistic treatment, not just, hey, one drug is either doesn't work or work and that's it, right? But also another very important part of what our platform is enabling. If we go back to our very first question, right? You might ask, say, look, we did all this sequencing everyone heard about, and why is that only helping 10% of the patient? Kind of the reason is right now, that's still very primitive, right? What we are looking at is, oh, do you happen to have a mutation that we have a drug to target, right? If not, move on, right? So that's all the sequencing of genomic information and RNA. Right now we have single cell sequencing. You have a lot of wealth of information, but it's being used in a very primitive way. Why is that? The reason is because you can imagine you get all this sequencing, look at all this patient diversity, but then the patient go out, everyone to have the exact same treatment, right? The standard of care. They can have first line, second line, maybe third line, then they're done, right? So basically you don't have the data sets to really take full advantage of all that sequencing to predict whether a patient is going to respond to a new treatment, right? That's ultimately the most important thing, right? But you don't have that training set, because every patient is going to have a FDA-approved drug, maybe one or two. They all have the same treatment, and that's it, right? So what this platform suddenly, as you can imagine, is suddenly providing the avatar platform that running virtual clinical trial, running actually, these virtual clinical trials that allow us to harness the power of all the genomics that we actually spent our whole society spent so much investment on. So what I meant is, right, in a recent study, for example, we showed everyone, we did a study, a virtual clinical trial, which is, we look at 1,000 past clinical trials. Most drug failed, some succeeded, right? That's 1,000 clinical trials on colorectal cancer, 215 drugs that came for those trials. And we basically now we're generating these patient avatars. For every patient, now we can test all 215 drugs on those patients, right? You're generating these big matrices, right? Before, your matrices is very, very one-dimensional, right? Kind of very thin, you can, right? You have 20,000 genes, and every patient has 3 drugs, right? So you can imagine the one you try to using AI to train. Sometimes I also joke, like a linear regression is good enough, right? Like you're not gonna with 3. But now with this tool, you can imagine now we have each patient avatar test on 215 drugs, right, from all the past 1,000 clinical trials. And that entire study was done in 9 days, right? 9 days, you finish what people did in the last half century with those 1,000 clinical trials, and showed, right? Look, first is the results correlate with patient outcome very well, and second is show that, right, about 85% of those drugs, if you don't know how to pick the right patient to respond to drug, they never had a chance going to a clinical trial, right? So again, but third is now you have this wealth of information data. You have the genomics for all these patients, and now you have the response to 215 all these different drugs, and now you can finally do the kind of much more sophisticated AI learning that we can do to help to predict. Say, look, we can harvest, right? We can harvest genomic information and making much more sophisticated predictor to try to predict whether a patient will respond to a treatment, right? So you're kind of providing the necessary training set for next level AI learning to be able to, really predict, taking the precision medicine to the next generation.
Yeah, I mean, it's infinitely scalable, and I can see the computational capabilities they apply. We're already have Zetascale. You don't need secret computing for this, but the fact that we do have exascale out there, and we're going to Zetascale, then really there's no limit, and it's because you created such a foundational methodology and platform and process that the applications are endless as well. I can see that. It's really amazing. I could literally spend hours sort of mining what you've done. We don't even have time to get into your work on this brain gut axis and the vagus nerve and all of this thing. And I think that's really interesting work as well. And so in the final minutes, I actually have a sequence of about 5 interviews today, so that's why we're going to have to start closing this one down. But, just fascinating. I know you started a company. How long did it take you from the formation of the company? And I know you were originally targeting to raise 70 million. You ended up extending it to 89 million Series A. How long did that take to do that?
So yeah, great. Yeah, just a very quick, right, kind of recap. So we formed the company in November 2019, right? And
that's incredible.
And yeah, so when I say immediately we got a Series seed. So our seed round is $5.4 million. And then in July 2021, last year, and so we kind of we went in and did this Series A, where started with we really going in and saying, look, right, we're gonna raise like $25 million and do this, but the investors, and we were lucky enough to really have strategic investors. So our Series A lead, right, including is Mubadala, which is the Abu Dhabi, right, the UAE kind of sovereignty fund, and they really kind of come in and kind of want to make a difference, and they also instantaneously recognize the potential of this technology, not just in the clinic, right, as a diagnostic, but also as a therapeutic, kind of platform for revolutionized cancer drug development. And we also have, managing partner of Google Venture, right? So Krishna Yashwan, who's really a legend in the field, and himself is a computer scientist and a physician, So and then also we have called EQT, which is actually the 7th largest private equity fund, and they have the largest European kind of life science VC, and another one kind of Two Sigma is the seventh biggest hedge fund, right? Who actually focus on data, and they really come in, kind of as you pointed out, see the potential of the data that we're going to be generating that no one else could. So we kind of have really visionary investor coming in, and then furthermore, interestingly is we have so when we start doing this, and again, the leading cancer centers like kind of MD Anderson also saw kind of the potential of this platform, and actually they kind of talk to our investors, and we increasing another 19 million, as you said, as extension to really kind of starting these pivotal trials and also platform studies with leading academic cancer centers around the world. And now our first registered trials, led by MD Anderson, but also joined by like Mayo, joined by the Cancer Core from Europe, and really coming together. And I think it's really a effort that the whole community sees that what's missing, right? Like we can keep doing incremental stuff, but how can we rethinking about cancer care? And by the way, the technology not just for cancer, right? It can model any tissue, like a fibrotic disease, infectious diseases, but kind of really kind of getting together and move to the next level. So again, we kind of really enjoying this kind of enthusiasm from our investors, but also from the key opinion leaders, right, in the clinical world, that coming together to make this a reality.
Well, I just want to sort of finish off with some, closing ideas and get your final recommendations. But, I should mention that you're a professor and chief scientific officer for the Terasaki Institute for Biomedical Innovation. Of course, you're founder and chief executive officer of Xiling Incorporated. Xiling being this company we talked about that you formed in 2019, and you've raised this 89 million Series A to advance medicine. You were the Hawkins Family Associate Professor in the Department of Biomedical Engineering and director of the Wu Center for Big Data and Precision Health at Duke University. We mentioned earlier you were scouted out of high school and got your bachelor's, master's, and PhD from Stanford, and you got the National Science Foundation Career Award at Cornell. We're on the steering committee chair of the NCI, National Cancer Institute Patient-Derived Model of Cancer Consortium, and co-chair of the NCI, that is the National Cancer Institute Tissue Engineering Consortium as well, and Cancer Track Chair of Biomedical Engineering Society 2019. So just sort of really fascinating that an electrical engineer moves into biomedical innovation, and you're doing just such marvelous work. Xiling, I'd love to get into your brain gut axis and the vagus work. I think it has really profound ramifications as well. We've got to do another interview just on that. But now it's back to you. Excuse me. Do you have any final recommendations you want to make to the audience?
No, yeah, it's great. I think I think oftentimes, right, like when we started to, people always think about, people base their past experience saying, oh, certain things are impossible, right? Like when we started this, and initially, people think, wow, that's so many people tried, how you guys can change that. And I actually had a chat with my investors. I say, look, kind of why you guys have so much faith, right, in academic professors that kind of really started doing this. I think they mentioned the three things that I think it's kind of really good takeaway. They say, look, right, we look at you and your co-founders, you guys have such passion, right, and your passion is not for just kind of making more money, but you guys are passion is for paradigm shifting and changing that. And the second is but what the reason we also invested is, right, you guys came in and kind of, we look at a previous technology, like the the patient-derived xenograft, right, the animals and stuff. But you guys can really identify the critical unmet need, right? And you guys have a solution for that. So that's kind of, and we heard so many talk, and people kind of end up always saying, look, right, this is the challenges. So although we hear around the field, we know this is a difficult problem, but second time, you guys are addressing the exact critical unmet need that we heard, right, 10 times before from all these companies. All the pharma come say, look, yeah, we just don't have a good model, right? We can predict, if we can have that, right, we will make things are changing. So you guys are, kind of addressing a fundamental problem. And third is also, I kind of appreciate, they say, look, we put in because, even you guys coming as professors, you have the humbleness that you kind of acknowledge what you don't know, right? And I think that's very important. For me, I think, without my team, and very early on, we had help, right, from veteran business side, people who, in the business came in and kind of really work with me to build a company, right, with a vision. Because if you just have a vision, but you don't have the execution, kind of power capability, and that's really 90%, right, of the game. I come in and sound all smart, but I think it's really the execution, building the right culture for the company that can really deliver. I think that's the key. But also, Stephen, to your point, I still have these academic appointments because I think it's very important to keep ourselves right grounded. So for me, I always coming up and say, look, right, everything I say right today, I tell you, these can stand the test of time, right? Everything we are publishing in peer-reviewed journal in very top papers, right, by key opinion leaders and experts in the field, and we have to stay true to ourselves because in the life science field, right, I think it's easy sometimes to overclaim things because it's so complicated. But to us, right, I think that credibility, which that's why we're working with all the top cancer centers, again, with farmers, because they kind of they trust us, right? When we say things work or don't work, they know we are not sugarcoating anything. So I think that's also very important.
Well, it's just remarkable. And, Xiling, I am just amazed. I can just see this. You're still young, too. So just a marvelous career ahead of you. Thank you for coming in today and sharing so many of your great insights with the audience in a way that makes sense. They'll understand it, all the implications of it. And we got to do more of these series. And I wish you a great day. So, and I should mention, we met at the World Innovation Summit for Health in Doha, Qatar, just a week ago. And when I looked at your work, I was like, wow, this is amazing. So thank you again for taking the time.
Thank you very much, Stephen. And thank you for the opportunity to kind of providing this big platform for me to kind of really convey the passion of what we are doing. And thank you very much.
Thank you for listening to the Brand Called You videocast and podcast, a platform that brings you knowledge, experience, and wisdom of hundreds of successful individuals from around the world. Do visit our website www.tbcy.in to watch and listen to the stories of many more individuals. You can also follow us on YouTube, Facebook, Instagram, and Twitter. Just search for the Brand Called You.